Okay, so good morning, everyone. So I hope you have watched the videos that I have posted last week during our asynchronous meeting. If you're going to recall, we started the video with why do we need statistics, okay? And in addition to that, you were given or discussed the so-called descriptive statistics, okay? And the other type of statistics that I gave at the first part of the video, okay? We're going to talk about that next week, okay? So descriptive thing and the tests. And you have different terms that you were introduced like replicates, mean, median, okay? And then you're able to get the mean, median, and standard deviation in the first part of the uh, video or in the first video that I gave you. Okay, so if you're going to look at this, everything can be done with the Excel. And then we also introduced to the Gaussian or normal distribution, wherein if you have the mean and the standard deviation, you could be able to determine the fraction of population under a certain condition. Okay, so if you're going to recall, the formula that we use there is C equals to the target value minus the average, okay, divided by the standard deviation. And you're going to need the C table, or again, you're given how to do this thing using Excel. And then we also discuss about confidence interval. I also showed there a video how to get the confidence interval, do the t-test, f-test, and the outliers, where we use the one is the grabs test and the other one is getting it using the, what do we call that? How do we get the outliers, which we're going to do before today, q-test. So what's the difference between t-test and f-test? T-test would use mean and standard deviation. F-test is exclusive for standard deviation, but both of them is trying to see if there's a difference between the two. So I'm going to have two problems that cover some of these topics today, and then I'm going to end it with the standard calibration, and then we start with chapter 5. We're going to talk about the exam schedule later on. Okay? So just to see if you understand that concept, aside from the assignment that I have posted Friday to Saturday, Saturday to Sunday in between time, okay, I have here some problems. So first here is confidence interval. So it says here, The gamma ray spectrometer orbiting Mars has found that the percentage of iron in the Amazonian regions of the surface of Mars is 14.6%, with a standard deviation of 0.9%. So this is based on the individual measurement. So what is the 95% interval size to one significant? So you are going to ask here, what is the standard or the confidence interval like that one to one significant figures? And you have to use your infinity degrees of freedom for student T because 341 is just so big. Okay? So if we're going to solve the answer, you are asked to do it using this. So if you're going to recall, what is this? Anyone? How do you describe this? That is your? Anyone? Hello?
That is the mean.
Okay, that's the mean. Okay? So technically what you're being asked here is just this portion here. So S is your what? Anyone? That's your standard deviation. N is your 341. Now where do we get the T? T is what?
It's a critical value of.
Okay, that's the critical value. Okay, and that is equals to, if you're going to look at here, so how many percent confidence intervals? So 95%. Okay, so base there, use the infinity. So you're going to put 1.960. So the way that you're going to do is you're just going to determine this one because that's the one that you're being asked. So you're going to replace it with 1.960 times 0.9. You don't need to convert this to fraction. Okay, and then 341. So try to get the value that you get there. So what do you get? What value is this equal to? Anyone?
I got 0.096.
0.096. But as you could see there, one significant figure. So you round this up to
0.1.
So this will end up as 14.6 plus or minus 0.1, or a range between 14.42, no, not 14.4, 14.5 to 14.7. So that's the confidence interval that you have. Okay? Question? If I'm not mistaken, I also have an example on how to do the confidence interval in one of the videos that I have posted last week. Question before we go to a set of examples on the next item. So this is not only t-test, although it says here t-test. As you could see, you will do f-test and q-test. So measurements of pH from the two Great Lakes, Lake Erie and Lake Ontario, were taken one afternoon. Here are the results. Lake Erie 8.07, 8.42, 8.33, 8.22, 8.35, 8.44. Now Lake Ontario, on the other hand, 7.76, 8.25, 8.36, 7.97, 8.20, 8.35, and 8.40. Now, based on your calculations at the 95% confidence interval, are the two pH means and standard deviations different or not different? So you're going to get their t-test and f-test. And then there's another question here. Are there any outliers in the given set of data? So you're going to view their q-test. So what's the first thing for you to do here? Anyone? So these are the type of questions that you may get in the exam.
Find the mean first.
Okay, so you find the mean. And in addition to that, you can find the standard deviation. Because if you're going to look at the thing that you get is what? You need to calculate the t-count. And in doing that, okay, you can do the one that has the S pooled, or you can do a shortcut that I have not did in the examples that I given in the video, but this is one of the data instead of doing the S pooled. So you have this, the absolute value difference between the two mean, and then divided it, okay, over S squared, or standard deviation squared, divided by N1 plus S2 squared, standard deviation of the second set. So that's what you get with the t-count. And then you compare it with the t-table. So try to get the mean and the standard deviation here. You can do calculator or you can do Excel. So you just input everything there and then get the average and then get the standard deviation. If I'm not mistaken, I think it's 832 here with the standard deviation of 0.13. Try to check. And for this one, I have here 815 and 0.24. So let's say we have that value, okay? So we substitute it here. So 8.32 minus 815 over 0.13 squared divided by, how many n do we have? 1, 2, 3, 4, 5, 6. Then plus 0.20, no, maybe I can just put them together. 0.24 squared over 1, 2, 3, 4, 5, 6, 7. So what do we have? Please calculate.
I got
What we-
1.6.
Hmm?
I got 1.6. Is that right?
Hit 1.6. How about the others? How about the others? What value did you get? The others?
I got 1.6.
Okay. So everybody got 1.6?
Yeah, I got 1.6 too.
Okay. So we have there 1.6. Now, how are we going to look now for the t-table? How do we say they're going to be different or the same? So if t-table is greater than, or if t-table is less than the key calculated, okay, then that is significantly different. However, if the t-table is greater than the t-calc, then it's not significantly different. So this is the value that you're going to compare it with. Now the question now is which values are we going to use here? Okay, so 95% confidence interval. So which one here are we going to use? So the next thing that we have here is we need to get the DF, the degree of freedom. And the way we define, okay, the degree of freedom is it's always what? Less than the given replicates that you have. And in this case, you have what? Two sets of replicates. Okay, so if you're going to look at the degrees of freedoms, usually in statistics, this is the number of observations minus the number of parameters estimated from the observation. Now the question, how many parameters do we have here? Do we have only one? We have what? 1, 2 set of parameters. So whatever the total value that we have here, we subtract it with 2. Okay. So whatever the number of observation minus the number of parameters. Now there are 2 parameters here. There's 2 sets of parameters that we have here. So we have 6 and 7 minus what? 2. And that will give us 11. So that means we're going to look at the value here. So it's either here or here, but closer to 2.2. Now, what can you say about the value here? Is this value tells us that they are different, or this value tells us that they are not different?
It is smaller than the critical value, so it's not different.
Okay, so since the t table is more than the t calculated, so there's no significance difference between the two. That's how you interpret this test. Okay, so unless it's greater than 2.228, okay, that's the only time it will be significantly different. So we solve now the t. So what's the next thing that we're going to do? We solve now the f. And in the f, it's just s squared over s. Now, our definition here of 1 and 2 is not the same as the one that we have here. Why? Because the numerator should have a greater value compared to the denominator. So in that case, it will be 0.24 squared divided by 0.13 squared. And that would be equals to what? What do you get? What's the F calc?
I got 3.4.
Okay, so 3.4. And again, you're going to compare it with the table. Okay, so the table value will determine if it's significant or not. So if F calc is greater than the F table, then it is significantly different. Otherwise, it's not. And you have to use also here the concept of the degrees of freedom. So remember, this is now your S1. This is your S2. Okay? So if we're going to look at this table, so this is what we have. So this is the S1 and this is the S2. So we need to determine now what is our degrees of freedom for S1 and what is our degrees of freedoms for S2. Now here, since you only have one set of parameters that you're going to use, you're just going to subtract the number of observations with 1. So this is what 1, 2, 3, 4, 5, 6, 7. So the degrees of freedom is 6. 1, 2, 3, 4, 5, 6. So the degrees of freedom is 5. So how do we look at here? What's the value that we need to compare with 3.4? Anyone? I let you what we could choose it, so I will know if you really understand the concept. So, but what is our F table?
Four 4.21.
4.21.
21.
As it like this?
2. No.
Huh?
421.
421. So if we're going to look where it is,
7 and 6. Is it what it is?
because if it's a 6
No, it's a 6.
and a 5,
Yeah.
this should be the one that we need, right?
It's not 7 and 6.
Hmm?
Why is it not 7 and 6?
What is our S1 here? Erie or Ontario? Convince yourself. What's our S1 here? This is the value that we use. So what's our S1? Anyone? So you convince it's Ontario?
Lake Ontario's the S1. So the
So that means that's a 6. because it has a 7. So 7 minus 1 will give you 6.
And S2 should be 5 because the
Yeah.
sample size is 6 and it's 6
1,
minus
2, 3, 4, 5, 6. Minus 1 is 5. So if we're going to look at the table, so the one that will meet between 6 and 5, because this is the S1, so 6 degrees of freedom. And this is the S2, 5. So wherever they meet, that's a 4.91. Not this one, because that's 7 and 6. But that's the replicates, number of replicates, not the degrees of freedom. Did you get that? Anyone?
Yeah.
Because that will be a deduction if you're going to compare it with the wrong value. Even if you say 4.21 is the one that you get, and it's still larger than 3.4, but that's not the one that you should compare your F-calc with. So you have there 4.95. Everybody know why is it 4.95? Remember, this is not number of replicates. This is degrees of freedom. And degrees of freedom is usually described as the number of observations minus the number of parameters estimated from the observation. So if that is the case, what can you say about the F-test results? You could say it is also not different. Analyze. Analyze why I make this an S1 and this an S2. Because the rule that you have when you do the F-test, the S1 should always be higher than the S2 value. Okay? Question? Before we go to the last one. Question before I erase everything here.
No.
Okay? So the last one that we have here, you're asked if any outliers are there in a given data set. So when we have an outliers, we're going to look at what values? The one that's either too high or too low. And then we're going to determine if it's an outliers or not. So what are the outliers here, the possible outliers here? This one and this one. And here, what are the outliers for Lake Ontario? I think this is the lowest, right? And the highest would be that one. And then we're going to do a Q test. So if we're going to do the Q test, we have gap over range. So gap is just your suspected outlier minus the closest value that it has over the highest and the lowest value. So the other one would be 8.07 minus, what's the closest to 8.07? 8.29 divided by So what do we get there? To the two values. Anyone?
The first one I got 0.243.
Any other value? How about the other one?
I got 0.595.
Okay. So what we're going to do now is we're going to compare it with the Q value. Okay, so you have 1, 2, 3, 4, 5, 6. So I have a table here. Now, if you're going to look at it, you have to be very careful. Now it says here what? Number of observations, not degrees of freedom. So that means you're going to compare it with 6. So these are the values that you need to compare. So what can you say? Are we going to retain those values or reject both or reject one of them? So 0.56, 0.625, 0.742, 420 at a different confidence level. So if we're going to look at it, all of them are, so 0.595, I think at 90 degrees Celsius, what we call confidence interval here, you're going to reject the lowest one. But at 95 and 99, you're going to retain the lowest value. So you see there 560, so this is higher than 6.560. So you're going to reject this at 90% confidence level. Okay, but the one that we're going to do is 95, so you're going to retain at 95. So let's go to the other one. So you have here 7.76 minus, what's the closest to 7.76? 7.97?
Professor,
Yep.
for the 0.243, do we also retain it or reject it?
You retain it because it's lower than the value that you have.
Okay, thank you.
So all you need to do when we have the test, you just compare whatever you calculated with the table. So if it's calculated is greater than the table, then that's the interpretation they're significantly different in the F test and the T test. In the Q test, that means you reject it. Okay?
Okay, thank you.
So you have here 7.76 minus 7.97. And then what's the highest and the lowest? 8.36 minus 7.76. And in the other one, you have 8.36 minus, what's the closest? 8.35 over 8.36 minus 7.76. So those are the values that you need to calculate. So what are the Q calculated or Q values that you get? So what do you get here? Anyone?
I got 0.35.
0.35. Now how about here?
Zero point 0.0167.
So 0.0. What do you have here?
1.6.
1 or
1.67.
2?
Oh,
So it's 167 or 0.02. So we're going to look at those value and compare it here. And this time, you have this value. 0.507, 0.568, 0.6880. So if you're going to look at this, what do you say? All those values are retained. So question. How are you going to interpret the two sets of value here? If you have a question like that, what can you say about the readings of Lake Erie compared to Lake Ontario? So we could say Lake Erie has a higher reading than Lake Ontario, but they're not significantly different. Okay? And we can also say that the readings that we have here are closer compared to the readings that we have here. Why? If you're going to look, the significant figures of these, the standard deviation for the first set is 0.13 compared to this one that is 0.24. Now, they are not significantly different because if you're going to look at the value that they have, they will overlap in one way or another. If you're going to look here, the highest value of this is much higher than this one, but okay, you have some values that each of them has, like 8.35, okay, 8.33. So that's within the values that you have here. So if you're going to compare it, you could say Lake Erie, maybe like this one. Lake Ontario would be like that. But in one way or another, they will overlap. That's the reason why they are not significantly different. Because I think in my example, I showed you there how two sets of values are significantly different. Okay? Now, the next part that I wasn't able to record in this chapter is the one calibration curve. Okay, so at least by this semester, you should know how to construct a calibration curve, because that's the first thing I will ask you when you do the first set of experiment in Chem 331. So we have a calibration curve, okay, when we prepare different concentration of standards, and it shows response of an analytical method to known quantities of analyte. So usually it looks like this. So different concentration and the signal and the calibration curve would look like that. So how do you construct your calibration curve? Okay, usually you prepare known analyte samples covering a range of concentration expected for unknowns. You obtain the corrected absorbance and you're going to plot the graph of corrected absorbance versus the quantity of whatever you measure. In the example that I'm going to give here, it is protein. Okay, so you have here an unknown protein samples gave an absorbance of 0.406 and a blank head absorbance of 0.104. So you are asked how many micrograms of protein are present. So if you're going to look at this, okay, to answer this question, you should also know the value of your materials here. So the one that you have here now is the Y, and you are asked for the X. So that means the M and B should be given. So I'm going to give you the M as 0.0163 and the B as 0.0047. Now the question now is the given value of Y 0.406 as is, or are you going to do something to it? So you might have a signal like this one. And you might get it like that one. So how are you going to solve for Y? Or X, not Y. So what do you need to do with the Y value here? Are you going to use 0.406 as is? Or are you going to subtract it with 0.104? Because there's a blank.
You subtract the
Okay.
absorbance from the you subtract the blank from it.
So you're going to subtract it with this one. And that will be your corrected
Corrected.
Y value. And then you have it like this. So the question now is, what's X?
I got 6.09 micrograms.
So this is equals to 6.09 microgram. So let's try. So if you're going to look at here, so whatever value that you get there, Y minus B divided by the slope. So usually that's the overall one. So 406 minus 0.104 minus 0.0047 divided by 0.0163. So you have there, what do you get? The X?
I got 6.09.
6.09. How about the others? Do you get any other value?
1.82.
1.82. The others? How about the others? Because I get different value.
I got 1.82 as well.
1.82 also. Because if we're going to do the calculation here, so we subtract this, so this will become 0.302. And then we're going to subtract it with the value that we have here. That's 0.0047. Right?
Yeah.
And we further divide it with 0.0163 to give you the X.
Yeah, I found a mistake in my math.
Yeah. I
I got
got
18.2.
So what do you get?
I got 18.2.
The answer is around 18.2. Ouch. So you have to be very careful. Sometimes the given absorbance is just a y, but if you have a reading of a blank that is not equals to 0, you have to subtract it with the blank. Okay? So if we go on further, if I'm not mistaken, all means of determining the concentration of an analyte is just by doing a calibration curve. You're going to do this at the end of the semester when you determine calcium and magnesium in experiment 10 and fluoride in experiment 12. So what you're going to do is you're going to prefer different concentrations of standards, and you're going to get the readings of those different concentrations. So for instance, in the calcium and magnesium, use the AAS. And from that, you'll be able to get, so if you have a different concentration, so you're going to get a different signal. It can be a direct relationship or maybe like that one, depending on the instrument that you use. Okay, so for instance, this one. So every time you do the calibration curve, you're going to get the slope of the line. And from that, that's the one that you're going to use to determine the value of your x given the y value. Okay, and in your calibration curve, the r squared is also needed to tell you how linear your set of values is. So the closest it is to 1, the better set of values that you have. Okay, so the way that they do it is the best fit line, or the one that we have discussed in the laboratory. Usually that is what? There's a term that we use, that the method of least square. Okay, so if you have a calibration curve like this one, you're given an unknown solution gives an absorbance of 0.621. What is the corresponding concentration of your unknown? So you plug that value here, and then since this is a minus, you add it with 0.00131 and then divide it by 2.0343, and then you should get the corresponding x value. Or you're going to look at 6.21 there, you plot it, so maybe it's around here. So that's two ways for you to determine what your concentration is. So around 0.312. So around that area. So that's how you do a calibration curve. Okay. If you're going to look at the quality of the results, as I've told you, it's the r squared that determines the quality. The closest it is to 1, okay, the linear is your calibration curve. Now, if it's not close to 1 like this one, you will see that the set of the readings of your standard is not really a straight line. So sometimes, maybe this one is not applicable as linear, you can change it to a log, and the curve might be a better fit for that. So the important thing that you need to do here is, in every experiment that you do, you're going to use a calibration curve. Now, if ever the value that you have is here, please don't do extrapolation. Okay? Extrapolation is not, we could say, reliable. So if you have a value that is outside the calibration curve, the best thing for you to do is to dilute that value until the value falls on the calibration curve. And then you just have to multiply it by 2. So that's what we have for this chapter. So extrapolation outside of range of standard should be avoided. So the best concentration range should be the one that we have there. So I'm not going to discuss this anymore, because all I want you to do is to know how to construct a calibration curve. And once you have a calibration curve, you should be able to determine the unknown. Okay? So you're given the Y value, you should be able to get the X value. So question before we go to the next chapter. Question? Before we go to this quality assurance and quality control. So this is more on, you are already doing the analysis. So you want to make sure that the data that you have, okay, makes sense. So what are the things that you need to remember when you do quality assurance and quality control? Okay, so let's say are those two terms the same? Because sometimes people just interchangeably use quality assurance and quality control. So what we're going to do now is, let's try to see if there's a difference between the two. Okay, quality assurance versus quality control. So if you're going to look at the quality assurance, it says here it encompasses a set of procedures designed to ensure the reliability of the laboratory analysis. Okay, so quality assurance, we could say on the other hand, as quantitative indications that demonstrate whether data requirements have been met. Okay, or the way that we do is what we do to get the right answer. That is quality assurance. It refers to the broader process that includes quality control, quality assessment, okay, and documentations of procedures and results designed to ensure adequate data quality. So we could say quality assurance cover quality control. So it's just like this is an overall aspect of which quality control is just one of the things that is being covered. Okay. Now, if you're going to look at the quality assurance, it's a process-oriented and focuses on defect prevention. So this is a process-oriented. Okay. So you look at, is the process good? Quality control, on the other hand, encompasses a set of procedures designed to validate the measurement quality. So if you're doing quality control, these are active measures that is taken, okay, to ensure the required accuracy and precision of a quality analysis. So if quality assurance is process-oriented, quality control is product-oriented and focuses on defect identification. So one would like to prevent defects. The other one identify what's the defect is. Okay? So let's say you have this set of data. So how are you going to say they're good? So you have 18 more results above 50 or 91 nanomolar concentration and 4 more results below 31. So this is just one sample, one what we call source. I'm just going to look at it. Now the certified range is just between these two. So how are you going to improve the quality assurance and the quality control here? So you have to look at what are the methods that they use where they get this one and compare it. What's the method that they use here? And you may ask, since this is one source, did they take the sample at the same time or they take it at different time period? Okay, so those are the stuff that you have to take into consideration to see why they're not different. They're just the same sample, but how come they have different results? Okay, so if you're going to look at the quality assurance, this is what we do to get the right answers. It refers to the full concentration range of practices employed to ensure that laboratory results are reliable. For instance, the raw data. What is the raw data that we have? That's the one that you record during the lab. Okay. Now that raw data will still be converted. So raw data is just whatever the mass of your sample, whatever the initial and final volume that you get. That is the raw data. Now the treated data that you have, that's the concentrations found by applying a calibration procedure to the raw data. What do you have for those who did experiment 1? Which set of data that you have there is treated data? Anyone? What is the treated data that you have from that first experiment that you performed? For those who performed last week, there's one data there that we can consider already as treated data. So the question is what it is. Anyone? From the 6 that performed the experiment? What are those treated data that you have? Anyone? So what are the raw data that you have last time there? When you weigh the coin, you have the mass, right? What else the raw data that you have there? The initial volume and the final volume. So what do you get from the two? The volume that you use. That is a treated data already. Okay? So that's the difference between the two. Raw data is what you see in the mass of the sample that you get from the balance. Raw data is what you see. What's the volume of the liquid initially in your burette? And what's the volume of the liquid in your burette after? Okay? You add some of the liquid to your sample. And then the results that you have is what is ultimately reported, such as mean, standard deviation, and confidence interval after applying statistics to treated data. So Here, the results that you have is already has undergone some calculations, has undergone treatment, maybe statistics in some of them. Okay, so before we go more with the quality assurance, I just want to show you something that might be of interest to you. Why do we need to do this quality assurance or quality control? This one. I don't know what iPhone is this, iPhone 6. So the first issue that they have with iPhone 6 is what? They have this so-called bend gate, because some of the iPhones is just bent if you put them in your pocket. Okay, so what quality assurance and quality control that they did not perform there? That's the defect that they have there. They didn't do this thing. Okay, so you could have their iPhone 6 introducing the Plex or the Boomerang edition. Okay, and then it's not only iPhone that has a problem. Samsung also during that time has this so-called burn gate thing. So all of a sudden, okay, the Samsung just got burned while being charged. So they have to replace this thing. So these are just some of the quality control and quality analysis, quality assurance that those companies failed to take into consideration. Okay. So if we're going to look at this quality assurance process, so these are the questions that you ask and the actions that need to be taken. So why do you want the data and results, and how you will use the results? So to answer that question, you have to write the objectives. So what is your objectives? Maybe if you are a Samsung or an iPhone personnel, you can say is, how good is my cell phone? Will it bend? Will it get burned? Okay. Or let's say if you want to analyze the air quality right now that is happening in the West Coast. Why do you want the data and the results, and how will you use it? So you want to say, I want to look at the air quality. So there are parameters that you have there. And whatever the results that you have there, you're going to use it. Okay. And then you have to have these specifications. So how good do the numbers have to be? So you have to write specification. At this number, the air quality. Hello, are you still there? I
Yeah.
think I got lost. I don't know why. My signal are fine, so I don't know what happened. Anyway, yes?
I was just going to say your signal was outside of the range of specifications.
Yep. So here, so we go again on the specifications that we have. So how are we going to use the number given to us? Okay. So for you to determine, let's say, the example that they have is the air quality in the
Wait, are you
West Coast
sharing
that's
your screen?
happening right now. Huh?
Sharing your screen?
I'm in my screen. Just wait. Yeah, I'm in my screen. Oh, I mean it's not. Oh, it's not shared. Okay, so
Thank you.
okay. So let's say we want to look at the air quality in the West Coast at this time. So you're going to do an analysis. Now, you have to set specifications. Okay, how are you going to set if the air quality is good, bad, or worse. So you have to put some specifications on that. And what will be your basis? Okay, so you have to put methods that will give you the numbers that corresponds to the specifications that you set. And then in addition to that, you have to consider how many samples are you going to use, how precise the method is, how accurate the method is, the selectivity, sensitivity, detection limit, robustness, rate of false result. So you are the one who will set that thing. Now, these parameters that you have, this is very important. These are the typical analytical methods parameter. In your specification, you also have to include the blanks. Fortification. So fortification, did you add any standards to it? The calibration checks, the quality control samples, and control charts to monitor the performance. Okay, so in specification, usually you write everything there in black and white. What are the analytical figures of merits? And usually this has something to do with the analytical method that you're going to use. They have a figure of merits of their own. So in this step also, you're going to write and follow the standard operating procedure. Okay. So in the specification, you just try to determine how good the number should be. What precautions must be used in the analytical procedure, and how should samples be collected and preserved. And we go one by one later on the different analytical figures of merits that was discussed here. And then assessment now. So this is the time we're in. You already have numbers that you collected, and you're now going to compare it with the specifications that you set. So were the specifications achieved? So you're going to compare now the data and the results with the specifications. You're going to document the procedures and keep records suitable to meet the use objectives. And you verify now if the objectives that you have met. Okay? So usually here in the assessment, okay, you're now trying to see what is your result. Did it meet what you wanted to have in the first place? So if we're going to look at the basics of the quality assurance, so if you're going to look at the objective here, you state the purpose for which the results will be used. And then in the specification, you state how good the numbers need to be and what precautions are required in the analytical procedure. Now, the most important thing that they said you need to do in analysis has something to do with the sampling. The strength and weakness of your results will depend on the what? Samples. So as much as possible, your samples is what? A representative sample. Okay? So how do we know, how do we say that our sample needs to be a representative sample? So one example right now, it's an election year. You may ask yourself, how reliable are those surveys? Okay? Because when you're doing the survey, you're not getting the whole population, right? What you get is just some representative of the population. Now, the question now is, how reliable are those representative? So what are those representative samples that we have? How reliable are those persons that you interview? Okay? Because surveys are not really reliable, because it's very hard when you're dealing with what? The samples that you have. What's the sample that you have in the survey? Anyone? Who are the one being surveyed? So that's the problem with when you use the human beings as your sample. Sometimes they're not really represent the whole population. So that's the thing that you do with the QA. How reliable is the sampling? So it has something to do with the sample that you have, or it can also have something to do with the sample preparation that you have. So there are instances that you can have what? A false positive. It says that the concentration exceeds the legal limit when in fact the concentration is below the limit. Or what practical false positive we can have? Anyone? Pregnancy. How do you know if your if a pregnancy test is false positively? If the result is false positive? Obviously, if the one you being tested is what? A male person. Why? Why you cannot say that the result is or cannot be positive? Because male person cannot be positive unless what? If you recall the lecture 2 that we have. When does the male person has a positive result in a pregnancy test?
It means he has cancer.
He has a testicular cancer. Okay. But I don't know if you heard the news last year. There was an athlete. Okay. He was tested for drugs. And you know what happened? The urine test for positive for pregnancy. And they found out, upon investigation, that the athlete used the urine of his girlfriend during that time. Okay, so that's the example of false positive. Says that the concentration exceeds the legal limit when in fact the concentration is below the limit. Or we could say it's the one that says it is positive when in fact it is negative. And false positive says the concentration is below the limit when it is actually above the limit. So this is in fact that should be positive, but it becomes negative. Now, I may ask you the question, which results you rather have? A false positive or a false negative? Let's say in this era of COVID-19. Come on, each of you, give your opinion. Do you rather have be a false positive, false negative? Or what do you say?
The false positive.
You rather be false positive?
Yeah.
Yep. The other? How about the other?
I'd rather be false positive.
False positive. How about the other?
False positive.
Okay. False positive, I would say yes. I would say I would rather have to have a false positive. And this case happened to one governor. I think the Ohio governor. So when the president was supposed to be there, he was tested positive in the morning. And then they found out in the evening that he was negative in 2 consecutive tests. Yes, at this time, you rather be false positive than false negative, because false negative, you're going to celebrate not knowing that you have the virus, and in your celebration, you're going to infect the other people. At least if you have a false positive, okay, you become careful, you isolate themselves, and you can say, oh, I'm not even asymptomatic, because asymptomatic is a positive result and they also have the virus. But as I told you, the testing that they have, especially the one that has the blood, okay, that gives you a false positive and a false negative results, because at the earliest time that it was applied, there's what? 50% probability you'll be positive or negative, which is not good, because that means the instrument is not reliable in the first place. Okay. Now, other quality assurance that you need to take into consideration, you have the method blank, the reagent blank, and the field blank, and then you have the calibration blank, and then the rinse blank. So there's really a lot of blank. But how do we define the blank? I think I discussed it last time. Blank is what? Everything except the analyte. So now you're going to look at the different blank. So method blank, sample containing all components except the analyte. Okay. Reagent blank, similar to method blank, but it has not been subjected to all sample prep procedure. So what does it mean? In the method blank, if you add acid to your sample, okay, because that's how you analyze your sample, you do it to a method blank. But the reagent blank, it just remained there, untreated. So we could say reagent blank are untreated blank. Okay. Field blank, similar to method blank, but it has been exposed to the site of sampling. So it is different also from the reagent blank, because reagent blank is just untreated blank. Field blank, you expose it to the site of the sampling. Calibration blank, you use it in establishing the analytical curve. So it is included in the calibration curve. So the idea that you have with the calibration blank is you want it to be 0 over 0, 0 and 0 reading. If not, it might have a reading, and what you do, the reading from that calibration blank, you're going to subtract from the reading of all samples. Okay. Now the rinse blank, so this is used to flush the instrument uptake system and nebulizer between standards, check solutions and samples to reduce memory interference. So the way that you do it this way is during the reading, you have there a blank, okay, that just try to make sure to clean up the parts of the instrument that was exposed with the sample. And after a while, when you put the rinse blank, it should give you the reading that is similar to your calibration blank. So those are the different blanks that you have. Matrix. So this is a Keanu Reeves thing. So matrix is referred to everything else in the sample other than the analyte. Okay. So it can be simple matrix versus a complex matrix. What's the simplest matrix that you can have? Anyone? Among the sample, what could you say is the simplest matrix that you have? Maybe your water. Okay? Because the water that you have usually contain what? Ions that you get from the pipes or the piping system. Now, if you have even a much better, let's say, distilled water, that's we could say the simplest matrix that you can have. But the matrix become more complicated if it is exposed to the environment. Okay, compare tap water versus water from the lake or environmental water. So which matrix is complicated, more complicated? Anyone? Can you say environmental water is the same in terms of complication matrix as the tap water? Hello? So which one has more complicated matrix? The tap water or the environmental sample.
The environmental would be more complicated, right?
Yep. So the environmental sample is the one that is more complicated. Why? Because they're exposed to different range of chemicals. Tap water is just what? It's just going to be exposed to the piping system. Other than that, none. The water, environmental water, it exposed to whatever the surroundings has. If it has a factory there, then it exposed to the factory. Okay. If there are some animal farms beside it, then it is exposed to animal farms beside it. So you have there this so-called matrix, a more complicated matrix. And sometimes this matrix, okay, would complicate analysis. So let's say if you want to determine the chloride system. So in the tap water, you're going to expect that it will contain chloride. But in the environmental water, the amount of the chloride will be affected by the other species that you have there. So when we say complicated, the more chemicals that the sample has, the more complicated its matrix is. Now, some of these chemicals can enhance or lower the signal of the analyte that you want to analyze. And that is the so-called matrix effect. So maybe in the tap water, you try to read, okay, the sample that you have by standard addition, you might have it like this. But for the one that is environmental sample, it can be like this one, or it can be like that one, depending on how the matrix affect the signal. Okay. In QA basics also, you also do this so-called fortification or spiking. So although this is not good in real life, so if you go to a party, somebody spike a roofie in your drinks, that's the term of spiking. The same thing in analytical method. So when you do spiking, you do fortification. So known quantity of analytes added to a sample to test whether the response to a sample is the same as that expected from a calibration curve. So what does it mean? The more sample you spike, the higher should be the signal that you get. And then in addition, you perform also test samples. So here, the quality control measure to help eliminate bias introduced by the analyst, knowing that the concentration of the calibration check sample. So what does it mean? From time to time, you have to put known or blank during the analysis just to test, okay, if the method that you use is working. So they said one thing that you're going to do if you end up in a quality assurance or a quality control work is you're going to do your work repeatedly, monotonously. So you're just going to do the analysis over and over and over again. And sometimes you become your work becomes not so challenging. So you develop some biases when you do the analysis. So how do they check that their quality analysts will do their work? So some of the supervisor, they put some known concentration. And that's the way they found if the analyst is doing his or her job. Okay? So he's just doctoring the results that they have, that he or she submitted. And then we have the standard operating procedure. So this states what steps will be taken and how they will be carried out. And usually this is part of the quality assurance, the bulwark of the quality assurance. And the way that you do this is later on is in terms of this chain of custody form. That's the standard operating procedure. So if you end up in forensic analysis, you have to monitor what happened to the sample that you collected as it came from the crime scene until it goes to the lab. Okay. Assessment, this is the process of collecting data to show that the analytical procedures are operating within specified limits and verifying that the final results meets the use objectives. So the chain of custody form. So I'm not sure if you're taking already your forensic science course, but this is the chain of custody form. So what does it tell us? It tells you the description of the item, where it is obtained, and who obtained it, and for what case. And then from that one, okay, you're going to see what happened to the sample that you have. So in every step that we have there, temporary disposition, release by, release to, temporary disposition, release by, release to, temporary disposition, release by, release to. So let's say if you're in the Olympics, okay, you collected the urine sample of some athlete. So what do you do? So every time you do analysis, you're going to put there, and then you're going to put, oh, stored in -20. Stored in -20. So every time you do an analysis, you're going to put it there. So they will know what happened during the time, because every time there is something wrong in the analysis. So let's say it was released and then you forgot to return it to the rep. So you compromise the result. And the chain of custody will see that. Okay? So the way that you look at this, the chains of custody is just telling you a story of the sample that you have. Okay? I'm trying to show you an example here where the chain of custody was used. What's the problem that we have with the professional sports? Anyone? What's the problem with the chain of in the professional sports that we have? Usually some athletes are what? What's the problem with some athletes in professional sports? They use performance enhancing drugs. Just like this one. I don't know if he's still playing, okay? But this is the a case that this player was able to win. And how did his lawyer able to overturn his case? So it happens here. Criticized drug testing as fatally flawed, citing the 44-hour log between when his urine was collected and when it was given to Federal Express. So how do you think they were able to know that 44-hour log? It has something to do in the chain of custody. Because if you're going to look at here, there's always this one. That's why if there's some missing R's in between them, you can question that thing. And he was able to win his appeal. But the second time, he got positive of using PED, he was suspended by baseball. Okay? So sometimes when they collect the sample, there are also analytical methods that's not yet developed. Who's that? The case, the cycling case, who always won the Tour de France, but they found out that he's dope. Is that Lance Armstrong? If you're following the sports, and you may ask yourself, how come it took that so long for him to be detected as using performance enhancing drugs, because it's the only time that they were able to develop a method to determine the trace amount of the thing. And there's another case that they have, and I think this chemist was already released from the prison. Okay. So what did she do? So what she do is whenever he do the analysis, okay, he always makes sure that it is above the level for conviction. So usually they said if you have a drug case, it's always on the amount. So I think the greater the amount, the harsh is the sentences that happen. So what happened? Okay. So it says here, they're trying to look at the one that contains THC. What is THC? That's the tetrahydroxycannabinoids. Okay. So what they found out is most of the sample that he analyzed, he spiked it with materials. And when they do the validation, which usually involves another person, they found out that there's always a difference. Those samples that pass by her, they contain fewer THC. And that's not good, because that means she's spiking the drug samples that is being collected from crime scene. And what happened here, look, 34,000 cases. Okay. So whenever you have a person who did a lot of analysis to the point that it is impossible, most likely that person is not doing his or her job. And what's bad here is because of the anomaly that he did, okay, they have to drop the cases wherein she was involved in the analysis. 34,000 cases in 9 years, that's just too much, I would say. So maybe because of ambition, okay, she was able to do it. But still, as I told you, okay, once you're caught on this thing, it's very hard for you to clear your name. So I think at this time, no lab would even willing to hire this person. So that's the thing that you do when you have this quality assurance. Okay. So in the quality assurance, it's just trying to make sure that you're doing the right thing that was given to you. So method validation. So as I told you, whenever you have a result, it doesn't mean that that's the result itself. Method validation, you have to do it to prove that the analytical method that you're using is acceptable for its intended purposes. Okay. For me in my lab, before we do publication, I want all results to be validated. How does it being done? If I'm aware that the method is already validated, what I want to do now is ask another student to do the same method, to use the same method as used by the previous student. If the results that they have are the same, then I would say it's valid. If not, what will I do? If student A has this different data from student B, what will I do? Maybe get another student, okay, and try to see which results are valid. Okay. The problem will come if student A, student B, and student C has different results. But usually what happens, 2 out of 3 students will have results that's close to one another. Okay. So in the method validation, you're looking at the 3 R's: repeatability, Okay, we start with the 3 R's: repeatability, ruggedness, and reproducibility. So repeatability is just the spread of measurement, and it's established through the standard deviation. So what does it mean? As much as possible, when you do it repeatably, the data that you have are close to one another. Ruggedness, so this is the assessment of precision or variation by different analysts using different instruments on different days, but within the same laboratory. So what does it mean? You do the same stuff, but you do it at different days. Because maybe during the colder days, you might have different result when you than the one that you did during the warmer days. It can happen. Okay? So that is ruggedness. And then you have reproducibility. So reproducibility is just precision. It refers to the variation obtained by different analysts using different instruments in different laboratories using the same analytical method with similar samples. So here now, you can tap another lab to see if the method that you're being validated is reproducible. So usually, you are now way here to the road of developing a standard method. Because you cannot have a method that's only reliable in your own lab. Okay? Whatever results that you got, it should be reproduced in another lab. If they cannot do it, then it can backfire to you. And there are many labs that happen when there is what we call, they question the reproducibility. I think one grad students in Colombia was caught doing that. When she left, just wait, she finished already her PhD, the people in the lab cannot reproduce the results that she obtained. And they found out, okay, that she manipulated the results. And in doing that, the advisor has to retract the papers that was already published. Unfortunately, they were not able to retract the degree that was given to her. Okay, but her name is already, we could say, she's already marked as one that is, what's the term that we could, who manipulated the data to come up with the result. So that's not good reflection. Okay. So in addition to that, other stuff that they use in method validation, selectivity, specificity. So how they differ from one another? So selectivity is ability of a method to distinguish the response of different analytes from each other. So means being able to distinguish analyte from other species in the sample. Specificity on the other hand, ability of the method to provide a response to a single analyte only. So this is the ability of an analytical method to distinguish the analyte from everything else that might be in the sample. So if it's selective, okay, that means even if there are other samples that you have there, you can determine it. Okay. If it's specific, it's able to determine this is chemical A, chemical B, chemical C in a given sample. Okay. So that's how the difference between the two. Linearity and sensitivity. So when we say linear, as the concentration of that analyte increases, the signal also increases to the point that you can do a calibration curve. Sensitivity, similar to linearity, but it has something to do with the lower level. So capability of responding reliably and measurably to changes in analyte concentration. So what does it mean? If you change the concentration, okay, you'll be able to see a change in the signal. But it has something to do in the lower one. You're trying to do how sensitive the method is to a certain analyte. So linearity, we could say when you use a higher concentration already compared to sensitivity. Accuracy, so nearness to truth. So the way that they do this in method validation is by spiking. So you spike a known concentration and you try to determine the so-called the percent recovery. That's how they do the thing. So what does it mean? If you spike a known concentration, you should be able to determine it when you analyze. So how accurate it is, usually if it's a 100% accuracy, then your method is accurate. Range. So not all methods when you develop can be applicable at different concentration. Some of them are just applicable in a certain range. So range is the concentration interval over which linearity, accuracy, and precision are all acceptable. Remember what I told you about the Beer's law? Like that one? So you could say your method is just applicable from this range. Over that range is not applicable anymore. And then we have the LOD and LOQ, which we're going to do today. So LOD is the smallest quantity of the analyte that is significantly different from the blank, while LOQ, that's the smallest amount that can be measured with reasonable accuracy. So usually this is what? 3 times the blank, and this is 10 times the blank. And we have a calculations of this in this chapter, how to get LOD and how to get LOQ. Now, robustness, okay? So it's a measure of the capability of an analytical method to remain unaffected by small but intentional changes to parameters in the method. So what does it mean? So you change the conditions, okay, on purpose. And if the result remains the same, so you could say the method that you have is robust. Okay, that's how they define robustness. So these are what we call the stuff that you need to validate the method. So once you meet everything here, your method can become the so-called standardized method. So how do you think they came up with this Beer's law? How do you think they came up with this method that they use right now in the COVID testing? Okay. So the problem that they have right now in the development of vaccine is they have to make sure that it is what? A valid one. So they have to do some method validation there. So how do they do the method validation there? They do a lot of testing. So what's the problem with the vaccine from what company is that? Pfizer? Some participants develop neurological something. Okay. I don't know if I'm going to ask you to take the vaccine, the first instance it will come out. Because we don't know what will happen. Okay. The results from animals is not the same as the results from human beings. So they have to do this what? Preclinical trials. And I'm not sure if a vaccine will be available by the end of the year. Although they already said that there's already one developed by China and Russia. But my question, are you going to take it? Knowing that it's not yet validated. Okay. I think Russia already said that they're available, but the questions being filled by the scientific community is how reliable it is. Did it pass through the safety level that is needed? Because I remember one time in England, they're developing some anti-cancer drugs, and all of the sudden during the clinical trials, one of the person that were given died because of the drugs. So what they do, they cancel the trial. Okay, because what will happen when they come up is it will be a Gaussian curve. So what does this mean? Either these are too sensitive and this one would be too resistant. It's always like that. But the question is how reliable it is. I don't know if you know the case of the hydroxy cut. There's an hydroxy cut before. Are you familiar with the hydroxy cut? Medical stuff?
Yeah, that was the old weight loss medicine, right?
Yep. Is still available in the public?
I think they changed the formula. Before, I mean, well, they had to because it was hurting. I don't remember exactly what it was, but it was once they actually tested it, they found out there was a lot of bad things about it.
Yep. I'm familiar on that because their program of operation was in Buffalo, and I was a PhD student in Buffalo for several years. So it was so popular, but they found out in the end that it's giving them some bad stuff. So rather than get sued, I think they stop, or they stop and then they change the formulation that come up again with another product. So that's how they do in pharmacy. Okay, you can hit big with one product, but it will take you years before it's going to be released. Usually it's 10 years for a medicine. I don't know if you're familiar in, I forgot the name, Lipitor. You familiar with Lipitor? It's a drug used in cholesterol. So they hit big with Lipitor. Okay, that's why they said if you want to be rich, you just have to find some problems that we have in the society and come up with the solutions out of it. So right now, the main problem is the COVID-19. So if you're going to look at the stocks, one of them, I think Moderna, is that, I'm not sure if that's the correct one, but it sounds like that. The stocks, when I looked yesterday, it's up 200%. That's the highest among the pharmaceutical company. Okay, so if you have a stocks on that company, so let's say you invest 1,000, so this, if it's a 200%, it is already what? 3,000 values already. Okay, so that's how pharmaceutical companies become rich. And they also said that usually the pharmaceutical companies, they have monopoly on that and they don't want to take it away. Let's say if you say you find a natural cure for diabetes, overnight you're going to kill $1 billion industry. And I don't know if you look at some ads, they always what? 99.9% effective. No one has ever advertised 100% effective. I don't know if you watch some of these commercial. Okay, you never seen a 100% solution. It's always what? 99.9%. And the reason for that is if it's 100%, so after they take the product, problem solved. So they don't have any more customer that will purchase their product. So you have to take that into consideration also. Okay, and that's the reason why the scientific community has a hard time right now to what we call to tell the people about it, because you know it's easier for people to come up with some question, okay, that sometimes is not easily answered by science. So let's look at these fortifications. So I have mentioned how do we determine accuracy? And the way I put it there is something to do with spiking. So fortification or spike. Now, how do you do that? So as I told you, it's something to do with the recovery. So recovery is the fraction of analyte that is added to a blank matrix before analysis that is actually measured by the method. So example that we have here is this one. You have an unknown and you found to contain 10 micrograms of the analyte per liter. So a spike of 5 microgram per liter was added to a replicate portion of the unknown. Analysis of the spike sample gave a concentration of 14.6. So find the percent recovery of the sample. So if we're going to analyze this one, so that means overall, how many do you have? So you have here the spike sample. So if you have a spike sample, this is this value, 14.6. But how many of this is unspiked? This one, right? 10. And the amount that you added is 5. So if you're going to get the percent recovery, you do the operation here and then multiply it by 100. And what do you get? How much was recovered?
%.
How many percent?
92.
So you have here around 92% recovery. So that's how you do recovery study. So the next thing that we have is, so this is some sort of the recovery studies that you have. So how do we get the LAQ and the LOD? Okay, the detection limit. So if you're going to look at the data that you have here, so you have a data that contains a blank and a sample. So if you're going to look at the reading that you have here, 50% of the area of the sample of the detection limit lies to the left here. So if you're going to look at this, 1% areas of the blank lies to the right of the detection limit. Okay. So the way that they look at this at the probability, so remember the Gaussian curve. Okay. So they define the detection limit as 3 times the S. S there is something to do with the signal that you have. Okay, so if you're going to look at the curve here, it shows the distribution expected for a blank and a sample whose concentration is at the detection limit. So the area of any region is proportional to the number of measurements in that region. So only 1% of the measurements for a blank are expected to exceed 50% here. Okay. However, 50% of the measurements of a sample containing analytes and the detection limits will be below the detection level. So the way that they determine detection limit is you're going to have different measurements in such a way, the way that you determine the detection limit is try to see how you can distinguish the signal coming from the blank and the signal coming from the sample So most of the time, the signal of your sample will be what? 50% in the area of the detection limit. And your blank, 1% of that will lie at least at the detection limit. So if we're going to look at this, so let's try to see how do we determine the detection limit. So these are the parameters that usually given when you try to determine the detection limit, or LOD, limit of detection. So the way that you do is you prepare a sample whose concentration is 1 to 5 times the detection limit. And then you measure the signal from several replicates, as much as possible. They said more than 7. And then you compute the standard deviation. And then from that, you measure the signal from the number of blanks and find the mean value. And you can put here the minimum detectable signal as the amount of the detection limit equals to the amount of the blank, or the signal for the blank, plus 3s. Okay? And the corrected signal is y sample minus y blank is proportional to the sample concentration. And you can do a calibration curve wherein this is your y, and then you have a slope, okay, times the concentration of the sample. And from that, you'll be able to get the detection limit as 3s over m. What do you think is the s? Anyone? What's the s? Anyone?
Standard deviation.
Okay, the s is the standard deviation. So this is a little bit tedious calculation, but you usually do it, especially if you try to develop a method and you need to determine the detection limit. Okay. So they said the LOD that you have there is just 3 times the standard deviations divided by the slope. So let's try to do this one. So signals from 7 replicate samples with the concentration 3 times the detection limit were 5, 5, 5.2, 4.2, 4.6, 6, and 4.9. Now, reagent blank gave values of 1.4, 2.2, 1.7, 0.9, 0.5, 1.5, and 0.7 nanoamperes. Now, the slope of the calibration curve for higher concentration is 0.229 nanoampere per micromolar. So find the signal detection limit and the minimal detectable concentration. So what is the concentration of the analyte in the sample that gave a signal of 7 nanoatmosphere? So what do you need to do here? You need to determine the blank and the standard deviation that you have for the sample. So SD for sample, okay, and then maybe the average of the blank. So try to do it. Try to get first that average for the blank and the standard deviations for the sample. Try to do it first, and then we will continue how to do the calculation. So I leave it to you, maybe for the next 2 minutes. Try to do that, and then we'll do the calculation. Were you able to get it now?
Yes. You want the first standard deviation for the samples?
Yep.
I got
What do you get?
0.56.
Okay, so 0.56 nanoampere. So how about the average for the blank?
The average for the blank, I got 1.3.
So 1.3, or you round it, I mean, we need at least 2 decimal places.
1.26.
Okay, so 1.26. Because if we're going to look at the signal detection limit, if we go from this one, so signal detection limit, y blank plus 3s. Okay, so y blank that you have there, that's 1.26 plus 3 times 0.56. So this gives you around
1.94 is what I calculated.
so 1.94. Is it 1.94 or I think it's 2.94.
Let me. Oh yeah. I see. I messed up.
You
Sorry.
add them.
Yeah. I dropped the 1.
Because 0.56 times 3 is 1.68 plus 1.26. So you have 294. So that's the Y detection limit. Now, the next thing that being asked here, minimum detectable concentration. So I have to go back here. So that's the one that we get, okay? Detection limit, so minimum detectable. So this is 3S divided by M. Now remember, they give you the M here. So that would be 3 times 0.56 divided by 0.229. So this is equals to
0.385.
3 times 0.56 divided by
Yeah, I'm sorry.
0.229. No, I get 7 something.
Yeah, 7.34. I'm bad at math. I'm sorry.
So around 7.3. This would be in the concentration that you have there, micromolar. So what does this tells us? So it tells us if you have a concentration like this, okay, you're going to have a signal around 2.94. So we could say this is your X value and this is your probable Y value. So the minimum concentration for you to detect a signal is 7.3 micromolar. And as you could see here, this is a little bit higher than the one that you use, but that's fine. Okay? Because the way that you did it here, you try to find at what concentration at least 50% will be with the blank and 50% will be as the detection limit. So now that you have that, we can now answer the last question. What is the concentration of the analyte in a sample that gave a signal of 7.0 nanoampere? So this is now Y sample minus Y blank equals to M times the sample. So what do you have here? You have this, which is your 7. What is the Y blank? That's the one that you have here, 1.26. The M is the 0.229 and the one that you're looking for. So you divide both sides by 0.229, what do you get? So if you have a signal like this one, that corresponds to how many micromolar? 7 minus 1.26 divided by 0.229. Anyone? What did you get? Anyone? When you do this operation, what did you get?
I got 25.1.
Okay, so now does the volume make sense? If you're going to look at the values that you have, so remember the detection limit that we have in terms of the concentration, 7.3. Okay. Now a 7.3 would give you what? 2.94. So if you have a 7, so that means the answer should be higher than 7.3. If you're going to interpret it, it's going to be like this one. So that's how you determine the detection limit. Now you may ask, why 3 times? Remember, the S is what? Standard deviation, right? So why do you want to make sure that the signal that you have is really the signal from the sample? You use 3 times because you want to make sure that it is about 3 times above the noise. So when the signal, they said, is 3 times as the noise, it is readily detectable, but still too small, okay, for accurate measurements. So remember, LOD is just detection. So what is the one that they have for accurate measurements? They have the LOQ. And what is LOQ? What is the number that you have for LOQ? It is what? If LOD is 3 times, LOQ is 10 times the noise that you have. That's the problem that you have in the instrument. Sometimes you're not sure if the noise comes from the sample or it's just noise. Okay. And Dr. Dye will discuss to you about that noise when you go to instrumental lecture. I think it's the first chapter. How do you distinguish the noise as coming as the signal that comes from the sample and not just noise? So you put some sort of a number to multiply. So if the noise, if the signal is 3 times the noise, you have the LOD. If it's 10 times, you have the LOQ. Now, not all analysis involves this so-called calibration curve. In some instance, you have to use standard addition. Okay? So in the standard addition, the way that they look at this is the signal is directly proportional to the analyte concentration. And you only use standard addition if you have this case. Remember the matrix that I have discussed to you earlier? Okay. So if we have a matrix effect, the change in the analytical signal is caused by anything in the samples other than the analyte. So whenever, okay, when the sample composition is unknown and you have a matrix complex, and that matrix complex can affect the analytical signal. So standard addition is very useful in forensics since the matrix is mainly what? Made up of other biological materials. So if you have forensics, what are the samples that you use? Blood, urine. So there are a lot of components or other chemicals that is present there. Okay. So the way that you do matrix effect is you use this formula. So what does this X means here? What does the S means here? Okay. So if you're going to look at the X here, the X that you have here, that is your sample. The S that you have here, that's the standard that you add. Okay. So the I that you have here with X, that's the signal coming from the sample. And this is the signal coming from the sample that was added with the standard. That's why the name that you have there is standard addition. Okay. Known quantities of standards are added to your unknown sample. So from the ratio and proportion that we have there, we will be able to determine how much is the original concentration of the analyte in your sample. Okay. So let's try to work one problem in the standard addition. So we have here serum containing sodium gave a signal of 4.27 in atomic emission analysis. Then 5 ml of 2.08 molar NaCl were added to 25 ml of serum. So the spike serum gave a signal 7.98. So you're asked now to find the original concentration of sodium ion in a given serum. Okay. So if we're going to look at the stuff that we have XI, SF, XF. And we also have, if you're going to look at this IX and IX plus. So what's the value of IX we have here? Anyone? So the original sample would have what value? 4.27 millivolts. And then when you added that thing, it becomes 7.98. Now the question, does this value make sense? Does this make sense that when you added it with the standards, the signal that you have there is higher? Yes or no?
Well, yeah, because you have more sodium ions.
Okay. Now, we can put one side as just 4.27 over 7.98. Now the question now is, how are we going to put the other side? Okay. So if you're going to look at this, this is the one that we're looking for. So we don't know xi. I'll just put here the sodium. Now, how do we get the SF here? So if we're going to look at the SF, so we need to take into consideration here, what's the initial concentration of the Si? So we have Si as 2.08. That's the original concentration. And then we get 5 ml of that. Spike to, what's the new volume? Anyone?
100 milliliters.
Excuse me. So it's 100 ml because you add 5 with 95. So this will give you 2.08 times 5 divided by 100. So I have 0.104. Molar. So I'm going to put this here. Now, the next thing that you have, so this is SF. What is XF? Now, the problem with the XF is we don't know the XI because that's the sodium that we're going to analyze. But we know it's 95 over 100. Now, the question is, why do we know it's 95? Because that's the sample that we have, the serum that we use. So we have like this. So we now need to get the concentration of sodium in our sample. So if we're going to simplify this one, 4.27 divided by 7.98. So that is equals to around 0.535. And if we're going to rearrange themselves, you will end up with what? 0.104 on one side, and then you have 0.535 Na minus 0.95, right? You follow? So what is Na plus? Try to get a whiteboard just to assist you. So if we're going to do it, so you end up with this one, 104 molar So how do we rearrange this thing? So we need to get the Na plus. So what is the Na plus that you get? Anyone? Five, 535. Hello?
I don't think I did my math right because I'm getting a negative number.
Yeah, I'm trying to find out where I messed up somewhere. But let's try to see if we get them all right. So we don't have this one. It is, let's say we put it at x. And this one, that's 0.104. And then this one will have 0.95x. So what do we do with the other one? So that's 4.27 over 7.58. So that is equals to 4.27 divided by 7.58. I get around, what did I get there? .27, 798. I got 535. So I'm trying to see. Oh, I think I know. I know where I messed up. I messed up the math. So this 0.535 should be multiplied on, not with that one, but with this one. So I end up with x equals to 0.535 times 0.104 plus 0.95x. That makes sense? Because the way is this become whole number, right? So the one that will cross multiply with that is this one, not that one. Or you can do a multiplication, right? You multiply this one here, where you end up with 4.27 times 0.104 plus 0.95x. And on the other side, you have 7.58x. So maybe you can start with that one. So please continue to get what's the x. So that's where I mess up in the math. So 4.27 times 0.104. So I get around 0.44 here plus 0.95 times 4.27. So I get around 4.56. Equals to 7.58x. So 7.58 minus the 4.0 and then divided by 0.444. Anyone doing that? Hello? Are you still there?
Yeah.
So try to do the math.
I got 0.126.
Okay. How about the others? How about the others? What did you get? Okay. So the X that you get there is around 0.1. So one get 0.126. How about the others?
I got 1.13.
1.13. So the starting point that we have is the one that we have here. Okay, X equals to 0.104 plus 0.95X equals to 4.27 divided by 7.58. Or 98, not 58. So that may be where everything's messed up. 7.98. 7.98. So I'm trying to look now. Minus 4.05. So 7.98 minus 4.057. So 3.923. So 0.444 divided by 3.923. The answer is 0.113. So if you're going to look at this, so what do you get here? 7.98 minus 4.057. So you get 3.923x. And you have here 0.4444. So you divide both sides by 3.923. You get around 0.113. So please confirm if you got it right. Anyone? Your assignment will be about that you're going to submit by Monday. Now, how about the other one that we can do? Okay, so we can do standard addition in the sense that you're doing a calibration curve. Unlike the one example that we have where we only have one set of sample in comparison to the standard. So when you're doing a calibration curve, it would be like this one. So this is the matrix that you have a groundwater, and this is the matrix that you have a reagent water. So when you do standard addition here, so you could say this is linear on this one, but this is linear on this one. So as you could see here, the matrix of the groundwater is more complicated than the reagent water. As seen here, you have lower signal compared to the one that has the reagent water. Now, how are you going to do it when you have a standard addition? So for instance, here, you place 5 ml, the same amount or volume of all the samples that you have. And you add different amount of the standards. Okay, the only one that you don't add is here. And then from that, you're going to dilute everything to 50 ml. And you will end up getting a calibration curve like this one. And to get the concentration of the unknown, okay, you're going to do it like this. And the y-intercept that you have there, that's the reading of the unknown without standard addition. And from that, you should be able to get the concentration of your unknown. So that is when you do a calibration curve with standard addition. But usually it's tedious compared to the one that we have earlier. Okay. Now, the other thing that they do is they use internal standard. So what's the difference of the internal standard with the standard addition? Now, in the standard addition, you're adding the standard that is similar to the analyte that you have. But for the internal standard, you're going to add another standard to the sample. So in the internal standard, you're adding known amount of compound different from the analyte that is added to the unknown. And then you're going to look at the ratio of the changes that they have. So the formula that you have here is this one. So we could say this is the signal of your analyte equals to a factor times the signal of your internal standard over the concentration of your internal standard. Okay? So internal standard is useful for analysis in which the quantity of the sample analyzed or the instrument response varies slightly from run to run. Okay, for a reason that is so difficult to control. For instance, flow rate. So even though you set the flow rate as 1 ml per minute, but sometimes they can go over or higher. So when that happens, okay, the signal may affect. So maybe you have a signal like this, and in another one you have a signal like this. But what you're trying to do is you're just trying to get the ratio of the internal standard with the sample or the analyte that you're looking for. Okay, so you also use internal standard when sample loss can occur in the sample preparation step prior to analysis. Okay, so the signal from the analyte is compared with the signal from the internal standard to find out how much analyte is present. So this is the thing that you need to remember. So let's try to analyze a problem that has internal standards. So in a preliminary experiment, a solution containing 0.837 molar of analyte and 0.667 molar of internal standard gave peak areas of 423 and 347. To analyze the unknown, 10 ml of 0.146 molar of internal standards were added to 10 ml of unknown, and the mixture was diluted to 25 ml in the volumetric class. So this mixture gave the chromatogram for which the area of the analyte is 553 and the area of the internal standard is 582. So find the concentration of X in the unknown. So you start with the formula there, RA over A over A, and then F RIS over IS. So the first step is just getting the F. Because if you're going to look at, you have what? 423 over 0.0837 equals to F. Then you have 347 over 0.066. So what is F here? F here, we could say that's your response factor. So when you do the calculation here, what is F? Anyone?
.96.
Is it 96? What's the number? 0.9?
6.
Is it 96? What's the next one after 6?
1.
1?
Yes, sir.
Everybody got that?
Yeah, I got the same thing.
So what do we do next? So what we do next is we're going to use that value, okay, for the next setup. So the setup that we have here now is 553 over, we don't know, equals to 0.96, and we have here 582, and then we're not clear. So we need to look at the A and the B here. Okay, so we could say this is your X, but your IS is what? So 10 ml of 0.146 added to 10 ml of the unknown. Are we going to put the 0.146 here? Yes or no? Is that concentration here the one that we're going to put here? Please answer me, yes or no?
No, because the volume is not 10 mLs.
Okay, so that 0.146, that's just the initial concentration. So we need to determine what is the exact concentration on that. So that means 10 ml over what? What's the final final volume that we have here? Anyone?
25.
Okay, so that's 25. So when we do that, so 0.146, okay, times 10 divided by 25. So you have here what's the volume that we have there? What's the concentration now? 10 over 25, so that is 40%. So 40% of 0.146. 0.146 times 0.1. So around 0.584. Did you get that?
What did you say? 0.0584?
0.0584.
Yeah, that's what. Yep.
So we can get the concentration now of the A here, but that is not the final concentration yet. So if you do the calculation here, 0.96 times 582 divided by 0.0584, so I have around 9,567 here, of which what you're going to do is you're going to divide it with that value. So I end up getting 0.0578. Is that the one that you get?
Yeah.
Hey, where are the others?
Just trying to see where I went wrong.
Yeah, I got something slightly different, but I think I have a rounding error. I think that's what it is.
The 0.961 times 582 divided by 0.0584.
Right.
So
For that.
I just used the whole 9,567 point something, and what I do, I divide it with 553.
Yeah, I just have a little rounding error. That's all.
Now the question is, how are we going to get the exact concentration? So if we're going to look at this, okay, so that's 0.057. What we're going to do with it is, we're going to look at what's the contribution of A. So 25 and 10 ml. So from that, we could say, I'm going to multiply it with 25 ml and divide it by 10. So that is the dilution factor, which is the reverse of the dilution. And if I'm going to do that, I'm going to get the concentration of what? Your A there would be equals to 0.144. Did you get that?
Yeah, I got that same number.
Yep, I fixed it.
Okay. So that's how you get the material. And if you're going to look at how you're going to see it in the chromatogram, this is how it's going to look like. So this is your analyte. And this is your internal standard. Okay? question? So we're going to change by next meeting. I don't want only a few of you participating. Or maybe we'll have our exam next week. So question.
And well, are the questions for the assignments and the exam, are they going to be posted similar to that, where they're given those exact values? And are we going to
Yes.
have an equation?
The equation is, it's up to you, because the calculation questions will be given to you at 9 p.m. and then you should be 9 a.m. and you should be able to submit everything by 12 noon.
Right.
So
Okay.
you will have your open notes for that.
Right. And I haven't checked, but the recordings of the of our lecture, they're going to be posted on Blackboard, right?
It's posted on the Blackboard, but I was told that they are only available for, I'm not sure. I'm not sure if it's 15 days or I don't know, but they told me that after that it will going to be removed because they want to have some cloud thing.
Okay.
So you have to make sure you can download the recording just to make sure that you have a copy of it. But from what I'm told, after 15 days, the recording will not be available because they don't want the cloud to be filled up with lectures. I mean, with the stuff of the recording, because it's a big file that they have. Now, for the exam, I found out that I think I made a mistake there, but if you're going to look at the mistakes that I have there, it was clear that I make it like asynchronous. Because I think what happened is this one, I put it on the 21st instead of the 28th. So the original that you have there is this one. But the way that I put there, there's the word asynchronous because on the 28th, we're not going to meet again. So I'm thinking, is everyone available on the 28th? Hello?
Yes.
You
How
said
about the others?
you said the 28th we're not meeting for class. Is it a holiday?
It's a holiday. It's Yom Kippur, I think.
Okay. And
But it's scheduled to be asynchronous, the one that was seen in the, where's that? I'm trying to look at the calendar of the PACE calendar. So if you're going to look at this, this is how it says there, Yom Kippur, remote asynchronous learning. But I want to make sure, because some of you may be Jewish and celebrating that thing, so I can consider that.
I will be available that day.
How about the others?
Oh my.
So-
Hey,
Available.
so who are not available?
His exam will be in the same time as class, usually.
Yeah. So I'm going to open the exam at 9. I mean, you're going to start working on it, and then by 12 o'clock, you should be done. And the thing is you have the calculation problem, and then maybe I'm not going to use the, what do we call that? The respondus, but you will be time pressured for the questions that are in the Blackboard. And you should be done by 12 noon. And that's the only thing that we're going to do that day.
Okay. I think I'm available that day.
So the first long exams move from the 21st to the 28th?
Yep. because
Okay.
it was really a mistake. Because
Okay.
the word that makes it a mistake is the keyword asynchronous.
Mm-hmm.
Now, the asynchronous time that we have is not on the 21st, but on the 28th. So it should be there in the 28th.
All right, I understand. Thank you.
Mhm.
And so the exam one, so it's not going to be anything covered for QA and QC, right?
For this one, no, I mean, QA, QC is now, it's more on statistics.
Okay.
Because we have 3 exams. So what happened is we're not going to do the advanced equilibrium. We're just going to do it in the later part. Anyway, we're not going to move to meet on that day. So it's just exam. But I want to make sure I'm not infringing your religious rights, especially if you're going to celebrate the Jewish holidays during that time. That's why I'm trying to know it as early as today. So any one of you going to celebrate that day, just let me know, because you will be excused from taking the exam, but you're going to take it at a time that you're going to set with me. So everyone is okay on that day?
Good to go.
Yes.
Mm-hmm.
Okay. And another thing that I have to look at is the lab, because as of now, I was able to get that there are 10 people who can do the lab, but I don't want the 2 people to be doing it the whole time. So I'm going to distribute the lab on when are you going to do it. Okay, so there will be week that both of you who are group mates, okay, you might be there. Because what I'm going to do as those 6 students who were there in the lab is you're going to submit all the raw data with me, and that's the one I've selected the raw data. That's the one that I sent to those who are not doing the lab. So maybe the other 2 who are forced to be there because the group mate is not cannot do the in-person, and the other one who have no partner, I expect you at least to be back this week. And then by next week, I'm going to already set the schedule, okay, of who will be the 6, because I don't want the other 2 to just be there the whole time. Because others told me they're willing to do the lab, even if they have the partner, okay? So I'm going to email the 10 of you, and we see this, how the schedule can be set. So for now, that's all. Now I already posted the quiz that you need to submit by 5 p.m. today. And then the assignment that's supposed to be for today, but I set it on Monday. And then I'm going to send also an assignment that you're going to set on Monday. So assignment 3 deadline will be on Friday. I think a minute before midnight. That's the one that I set there, 11:59 on a Friday. I cannot meet you today for office hours because I have this university committee meeting. I have 2 meetings after today. After I meet you, starting at 12 until 3 o'clock. Okay. So question.
No.
No.
Okay, now then we'll see. I'll see some of you on Wednesday. Okay, so stay safe and see you on Wednesday.
Professor, I have a question.
Okay.